Our next speaker, still in our session 3 on nanomedical structures and devices in development today, is Dr. Shuyi Li. Dr. Li is at the Institute for Molecular Medicine and Genetics at the Medical College of Georgia, and we're very pleased to have him speak to us today on a topic which is absolutely essential to achieve most of Ray Kurzweil's visions for the potential of medical nanobots, namely modifying the function of DNA repair nanomachines for therapeutic benefit. Dr. Li.
Good afternoon, everyone. First, I would like to thank the conference organizer for inviting me and giving me this opportunity to present my work. And also, I would like to thank the Genesis and the Temple for their great work. Actually, I came here, my first flight was canceled. Oh. And then I took taxi to Atlanta, and then I arrived in Montreal last night. To be exact, midnight, around 12, and I just arrived here this morning at 2 o'clock. Even though I sleep only 3 hours, I'm still okay. I mean, because this meeting is very stimulating.
Thank you. Thank you very much.
Okay. So I'm from Augusta, Georgia. Actually, our institute is part of the NIH Nanomedicine Center for Nuclear Protein machines. This center is included several institutions, including Medical College Georgia, Georgia Tech, Emory, and Cold Spring Harbor. So today, my talk is divided into 2 parts. First, I will briefly introduce the mechanism of the DNA double-stranded breaks. I will explain why one particular kind of damage is much more potent than others. In the second part, I would like to provide some evidence, some proof of concept experiments. We are develop a therapeutic antibody. It's called single-chain antibody fragment. By folate receptor-mediated endocytosis, we deliver this to the cancer cells. We hope that we can use this to manipulate the cell response to ionotic radiation. Actually, we got some evidence. I will show everyone here. So first, I would like to mention what is a nuclear protein nanomachines. In my understanding, the nanomachine at least should include the following components. It's naturally occurring nanometer scale structures made of protein and nucleic acid. Generally speaking, this machine usually contains about 10 to 200 components. They carry out specific functions such as information copying, transfer, and maintenance. 2 of our examples are DNA double-stranded break repair machines. Another is RNA synthesis machine. If you see that these 2 machines, they have certain similarities. First, they using the nucleic acid as the raw material. And once they recognize specific sequence, they assemble it. And then they get their product, they forming the phosphodiester bonds. Generally speaking, the nanomachine, it's a very complex controlled mechanism. Our center, we are trying to study the DNA double-stranded break repair nanomachine. The reason why we choose this as our model system is that because DNA double-stranded break repair machine is well characterized, and it's evolutionally conserved. There is a clinical relevant. So the biological question is how cells maintain their genome integrity. Each cell genome, they serve us about 17 to 100 years. But unfortunately, each cell, they suffer about 1 million DNA damage events per day. So inside cell, there is sophisticated DNA repair machineries. If there's anything wrong, it will cause some big trouble. Many genes that previously thought to be interfere with lifespan has turned out to be DNA repair genes. So nowadays, we found that there are about 150 repair genes. There are about 12 types of nanomachines. Among the DNA damage, DNA double-stranded break is the most toxic damage because unrepaired DNA lead to cell death, and misrepaired DSBs lead to chromosome aberration, as you saw here that there are certain breaks. So let me introduce the source of DNA double-stranded break. So when the high energy of photon or other particles pass through the tissue, it will generate reactive species along the nanometer scale track. When the radiation track passes through the DNA helix, they may intersect with both strands. It will cause double-stranded break. In the RNA irradiation is a principal cause of the double-stranded break. There are three sources. For example, it's terrestrial, and every year people may receive 30 milligrams. Cosmic, maybe 26 milligrams. Another major source is anthropogenic. For example, when you have an X-ray examination, if you take the X-ray chest, you may get 6 milligrams. And if you have a GI tract X-ray examination, you may have 200 to 400 milligrams. Another major cause for the RNA irradiation is from the radiotherapy. Each year in the United States alone, there are about more than 700,000 people receive radiotherapy. For each individual, they may receive 60 to 80 gray. Another source for the double-stranded break comes from the chemotherapy reagent, such as topoisomerase poisons. Biologically speaking, within cell, there is another source is called VDG recombination. So our goal is that we want to develop tools to visualize the DNA repair dynamics in living cells. We develop bright, photostable probes. For example, we have established a different fluorescence conjugated fluorescence DNA repair or DNA repair pathway proteins using different colors. We conjugate different dyes to the DNA repair proteins. We try to visualize the repair process within cells. We also contribute to the quantum dots. In terms of the control DNA formation, we have developed the macro irradiator, try to just introduce 1 or 2 bricks in nucleus. We also deliver the zinc finger nucleus to the nuclear to the cell nuclear, and to get some cell get some break. We are using the high resolution of microscope, using the structure illumination microscope. This how I can show their movie.
It's not running, it wasn't.
Okay, that's okay. So within cell, the major pathway to deal with double strand break at the clinical relevant dose is called non-homologous end joining. So once DNA break, double strand break occur, the cell will detect, okay, there's something around there. And for example, you got break here, and then you will have the initial complex that is Q80 and Q70 will directly bound to DNA ends. And then this is a crystal structure of the Q protein. Q protein is a heterodimer of Q70 and Q80. This is a DNA within this heterodimer. And then it will recruit another protein, very important protein. It's called DNA-dependent protein kinase catalyst subunit. So these three will forming the complex. DNA-PKCS will phosphorylate itself and to other proteins, and then it will disassemble from their site. This is critical structure of the DNA-PKCS. As you can see here, that DNA is within these molecules. And then it will join together with other part, and finally, other proteins like XRCC4 or ligase4 will form the enzymatic processing and ligation, so the double strand break can be repaired. Here is our radiosensitizer concept. So once cell get ionizing radiation, they will form their DNA-PK initial complex, and in their normal situation, the cell, once they get activated form of the DNA-PKCS complex, double strand break can be repaired. The cell, cancer cells still can survive. But if we can deliver some reagent like a single-strand antibody to interfere this pathway, we should be able to radiosensitize cancer cell to ionizing radiation. So I have cloned the single-strand antibody fragment variable. We know that the regular IgG composed of the heavy chain light chain. Actually, the antigen binding site is only the variable is here. So by the molecular technology, I have cloned this gene, and I also add a flexible linker here. This is a heavy chain light chain, and you can see here that DNA-PKCS binding site is on the top. There is a disulfide bond in each of the heavy chain light chain. Actually, part of this work has been published in the Nucleic Acid Research as a cover picture. So we first do the epitope mapping. So we want to find out where this therapeutic antibody bind to. So we found that this single antibody bind to the 25 residue, almost exactly in the middle part of the DNA-PKCS. So this is far away from kinase domain. So this is a regulatory domain for this protein. This is a kinase domain from EM structure. This is a kinase domain. Our antibody binding somewhere here. It will interfere with conformation, so interfere with the repair process. So first we perform the cell-free. Our lab, we established the cell-free system. Basically, in the test tube, you add linearized DNA, which is mimic double-stranded break. You add certain DNA repair components, like nucleic extract, which contains Q70, Q80, and DAPKCS. You add DNA ligase 4 and XIC4. So the linearized DNA can form the dimers and a high order of oligomers. If you add our antibody to this system, this antijoining will dramatically inhibit it. If you add a control unrelated single antibody, it still can form a ligated product. We also have a positive control that is non-specific PS3 kinase inhibitor. So suggesting that our antibody can inhibit the antijoining in cell-free system. So next, we perform the small colony formation assay. We macro-inject our antibody into the cell nucleus together with EGLP plasmid. GLP green here that help us to trace the fate of the inject cells. So in this column, both cells has receiving either our therapeutic antibody or control antibody. 88 hours later, the cell can proliferate normally. Same thing happened for the cell that's received the control antibody with sublethal dose only 1.5 degree irradiation. The cell still can proliferate. But on the contrary, the cell that receive our single antibody 80-2, the cell disappear. So suggesting that combination of the radiation and the single antibody 80-2 inhibit cell growth. So we next detect, actually we found about 60 to 80% cells disappear from the plate. For those cells that remain on the cup slips, we perform the immunostaining. We stain the cell with active form of caspase-3, which is a marker of the apoptosis. We found that the remaining cells on the cup slip, they show positive staining, suggesting that the cells are through the apoptosis pathway. So all the controls shows negative here. So we next test whether it's through the inhibition of DNA repair pathway. So we have a very sensitive marker. It's called gamma H2X. Gamma H2X is a histone variant. Once double-stranded break occur, it will phosphorate at a serine 139. If you stain it with antibody, you can see at 30 minutes at 1.5 degree, you will see there a lot of red spot. I will show you high resolution later on. But basically, information here is that at 90 minutes, for the cell receive the single antibody 80-2, the gamma 2X foci still persisting. The cell did not receive this antibody, they can recover, suggesting that the combination of the radiation and 80-2 can inhibit DNA repair. Our antibody can inhibit the repair process. So how can we deliver this antibody to the cancer cells? So first we choose the lung cancer as our first target because the lung cancer is the most common cancer both in men and women with a 5-year survival rate only 15%. And also radiotherapy is commonly used, almost used in this patient. But unfortunately, the local control is limited by the sensitivity of normal lung tissues. So the strategy that we are taking, we are trying to enhance the intrinsic sensitivity of tumor cells to RNA radiation. We also believe that this single antibody may also have potential as chemo reagent in, the sensitizer to enhance the chemotherapy. So how do we deliver this? We are taking advantage of the folate receptor-mediated endocytosis. Folate is overexpressed in many cancers, including lung cancer, on the cell surface. We conjugated our single antibody to the folate and bind to the folate receptor. It will form the endocytosis. The previous study has shown that the high affinity binding, even 0.1 nanomolar binding, will trigger the undestructive endocytosis. And also, there are some literature has so successfully delivered different size of protein successfully. So we using this strategy. So first, we want validate whether we can successfully deliver our antibody to the cell nucleus or not, because DNA-dependent DNA-PKCS is a function in the cell nucleus. As you can see here, that the cell that incubated with our folate conjugated single antibody, actually we have made a new version of our single antibody with a nuclear localization sequence. We're using the folate conjugated one, try to internalize to the cell membrane. So we use the nuclear localization sequence to help the nuclear specific targeting. As you can see here, that this is a staining for our antibody have a tag. This staining suggests that our antibody both distributed within nucleus and cytoplasm. The green channel stands for its target, DAPKCS. The merged yellow one shows that it's well colocalized with the nucleus. This is a double staining suggesting the nucleus. Can you use the live image here? Doesn't work? Okay, that's okay. And this is a live image for actually we conjugate our FITC to our antibody. This is lab cells, but unfortunately we cannot show the movie. But clearly the cell nucleus has been, the antibody has been internalized to the cell nucleus. And how does antibody work? We have performed immunostaining in this panel this is a cell that received the control protein. This panel received our therapeutic ADS2. As you can see here, at 30 minutes, the cell, this is a marker of DNA bricks, so screen. But at 90 minutes, the cell that received control protein, the green signal is disappeared, suggesting, the break has been repaired. This is a phosphorescent marker at a serine 2056, which is very near to the epitope binding site of our antibody. As you can see here, that it's both the red staining. But interestingly, if you see the cell that received our single antibody, at 90 minutes, there are still a lot of green signal there, suggesting that DNA damage still didn't fix there. But if you compare this to this, there is a dramatically reduce of the phosphorylation. So we believe that the mechanism of our antibody is at least by inhibition of the phosphorylation of the DAPKCS at this site. There may be some other site we are now trying to investigate it. So in the radiation field, the most golden experiment is to generate a cell survival curve. So first we are generate cell survival curve. We show here that if we increase the dose of our therapeutic antibody to the cancer cells, it shows dose dependent, suggesting that there's some killing effect. And we are also using different dose And this is our ICFV. This is control protein. I should point out that this is log scale. I mean, even though you see that there's not that obvious, but statistically speaking, there's big difference between these two groups. And also in the clinical setting, usually the dose is 2 gray. At 2 gray fraction treatment, you will see the big difference between these two groups. So how we can apply this to the future application? So one, okay. Let me start here.
You going to? Okay. If you want to go to the last.
Oh, okay. Excuse me. I just want to point out that
Yeah,
I think our principles are whether I just show one of the examples. Actually, we are now taking some other steps. So, I mean, we are using some gain of the gene correction. So we are deliver other zinc finger proteins and also DNA template to the cancer cells. And we will specifically carting DNA at a specific site. Actually, I just mentioned one of the pathway, no homologous joining. Within cell, there's another pathway, is homologous recombination. So we hope that in the future we can correct gene by specifically by this technology. And also, there are certain similarities. If we are able to find certain common features of the different nanomachines, we should be able to design the novel nano machines and to interfere or to solve certain biological problems. So finally, I must thank those who have finished this project. Actually, this is our Georgia governor, Sonny Perdue. He issued me the award for our research work. Thank you for your attention. Sorry, I kept too long.
No problem. No, your research was well worth the time. So no problem at all. Would people like to approach the microphone to ask Dr. Lee any questions about his research? Yes, thank you.
Alex Travis from Cornell University. This is getting a little outside my area, but there's a gain-of-function method where you can insert a gene for herpes simplex virus, thymidine kinase, and then render cells susceptible to antiviral drugs. And have you or anyone in your field considered that approach to, with a gain of function, actually causing an ablation of those cells?
Not this moment. Actually, our center, we have some expert in this field. Our very near future, we are taking advantage of homologous recombination pathway. We deliver the normal template and also the specific zinc finger protein to the cell nucleus and cutting specific site and using the homologous recombination pathway to correct the genetic errors. We hope to fix that genetic order within cell.
Okay, thank you very much, Dr. Lee.
Thank you.